मैं राजवीर सिंह शेरान एसोसिएट प्रोफेसर इन केमिस्ट्री SRR मोरारका गवर्नमेंट PG कॉलेज झुंझुनू। फर्स्ट ऑफ ऑल गुड आफ्टरनून टू ऑल द स्टूडेंट्स ऑफ MSc फाइनल। पिछले वीडियो लेक्चर के अंदर अपने सेल मेम्ब्रेन के बारे में डिस्कस किया था। टुडे वी विल स्टार्ट इन द सेम यूनिट आयन ट्रांसपोर्ट थ्रू cell membrane. Means substances are transported through cell membrane by either primary active transport or through secondary active transport besides diffusion transport of ions by these two mechanisms as discussed below. First of all, primary active transport or sodium plus and potassium plus ions. Substances transported by primary active transport include sodium, potassium, calcium, hydrogen, chlorides and few other ions. But all these ions are not transported actively through all the cell membranes. In some cases, transport occurs through intracellular membrane rather than the surface membrane, such as membrane of muscular sarcoplasmic reticulum. Active transport mechanism studies are mainly based upon sodium-potassium pump, a transport process that pumps sodium ions outward through the cell membrane of all the cells and potassium ions inwards. This pump is responsible for maintaining sodium-potassium concentration difference across the cell membrane and also for the maintenance of a negative electrical voltage inside the cells. This pump is also responsible for transmittance of nerve signals through nervous system. Functioning of this pump is discussed below. The carrier protein in the cell membrane is complex of two globular proteins, large one of which is alpha subunit of molecular weight approximately 1 lakh, smaller one is beta subunit of molecular weight about 55,000. The function of smaller protein is probably to anchor the protein complex in the lipid membrane. Alpha protein is responsible for sodium potassium pump and its main functions are: it has two receptor sites of the potassium ions on the outer side. Inside the inside portion of this protein near the sodium binding sites has ATP phase activity. When two potassium ions bind to receptor on outer side of the carried proteins and three ions of sodium attached to the inside binding sites of sodium ion, the ATP as function of protein becomes activated. Now one molecule of ATP splits to give one molecule of adenosine diphosphate ADP and have high energy of phosphate bond is liberated. This energy causes change in the conformation of carrier proteins, as a result of which these sodium ions get released to the outside of cell membrane and two potassium ions are dropped in the inner side of the cell membrane. This way, interchange or active transport of sodium plus and potassium plus ions across the membrane takes place. However, the exact mechanism of conformation change is still not known. Importance of sodium and potassium pump. Most important function of sodium potassium is to control the volume of the cells. Without pumping mechanism, most of the cells will swell up and ultimately burst. Inside the cells are larger number of proteins and the other organic molecules which are not move across the cell membrane. Their higher concentration causes inward movement of water by osmosis, as a result of which cell may swell up and ultimately burst. But as a result of sodium plus and potassium plus pump, 3 Na plus ions move outside and 2 K plus ions inside. Also, membrane is less permeable to sodium plus ions than K plus ions. So, once sodium plus ion move outward, they have tendency to stay there. This represents a continuous net loss of ions to outside the cells, which initiates the osmosis of water outside as well. Thus, if a cell begins to swell up for any reason, sodium potassium mechanism is automatically activated, move more ions to pump outside and also activate reverse osmosis, saving the cell from any burst, and this process maintain the volume of cell. Another important function of this sodium potassium pump is its electrogenic nature. As a result of sodium potassium pump, three sodium ions move outside the cell membrane for inward flow of two potassium ions. Thus, for one pump, one positive charge goes outside and it brings one negative charge inside. Thus, an electrical potential is generated across the membrane, which is basic requirement for nerve transmission. Primary active transport of calcium ions. This is done through the calcium pump, and calcium 2 plus ions are normally maintained at extremely low concentration in the intracellular crystals, cytosols of all the body cells. At a concentration of about 10,000 times lesser than the extracellular fluid, during its pump mechanism for calcium 2 plus ions carrier, proteins penetrates the membrane and served as ATPs, have capability to cleave ATP like sodium potassium pump. The difference is that this protein has highly specific binding sites of calcium 2 plus instead of sodium or potassium ion. Primary active transport of hydrogen ions. At two places in the body are primary active transported system for H plus proton in the gastric glands of stomach, and secondly in the late distal tubules of cortical collecting ducts of kidney. Deep lying Varial cells of gastric glands helps in transport of H plus ions by any part of the body. At the secretory sites of partial cells, H plus ion concentration is increased and they get released in the stomach where they associate the chloride ions to form hydrochloric acid. Now, energy requirement for primary active transport. In active transport of ions, energy is required, which depends upon the extent to which substance is concentrated during transport. Energy required to concentrate 100 folds is twice the amount required to concentrate it 10 folds, whereas energy is required to concentrate its 1,000 times is twice as compared to concentration its 100 times. Therefore, it may be concluded that the energy required is proportional to the logarithm of the degree to which the substance is concentrated. This relationship is expressed as follows. Energy in calories oblique osmoles 1400 log C1 upon C2. Energy required to concentrate 1 osmole of substance 10 times is approximately 1400 calories and 100 times is 2800 calories. Thus, the tremendous amount of energy is required to concentrate ion cells in the transport of them. Glandular cells like renewal tubules of 90% of their energy for this purpose. Secondary active transport. As a result of primary active transport, concentration of sodium ions outside the membrane become higher than the inside membrane, and a concentration gradient is developed, as a result of which sodium ions always attempts the diffuse inside. Under appropriate conditions, sodium ions diffuse inside along the sum of their substances by the process known as co-transport, which is a type of secondary active transport. Like this, sodium channels 0.3 to 0.5 nanometer side are negatively charged, which pull charges sodium plus ions into the channels and pull them away from hydrating water molecule, which diffuse in the either direction according to the law of diffusion. Thus, sodium channels is selectively for sodium ions. On the other hand, protein channels selective for K plus ions of only 0.3 nanometer size but are not negatively charged. Hydrated potassium ions are smaller than the sodium ions to a considerable extent because sodium ions attract more water molecules than the potassium ions. Smaller size of K plus ions easily pass through small protein channels selectively. Similarly to active transport, diffusion through the protein channels involves conformational changes in the shape of the carrier protein molecules. In case of sodium, channel gate opens and closes at the end of the channel on the outside of the cell membrane. Opening and closing of the mechanism. Opening and closing is controlled in two ways. First, voltage gating. According to this mechanism, molecular conformation of gate responds to electrical potential across the membrane. A strong negative charge inside the cell membrane causes the outside sodium gate to be tightly closed. Conversely, negative charge is when lost gate opens suddenly and allows the tremendous inside flow of sodium ions. This is the basic cause of action potential in nerves that are responsible for nerve signals. Potassium gates are on intracellular ends of potassium channels. They open when inside of cell membrane is positively charged. Opening of this gate is responsible for terminating the action potential. Secondly, chemical gating. Few protein channels opened by combination with chemical substance called ligands, which causes conformational changes in protein channels as a result of which gate opens or closes. This is known as chemical or ligand gating. Now, nerve tissue and nerve conduction. Nerve tissue, before going in details in neuroinduction, it is appropriate to know the structure of nerve tissues, which consist of neutrons. Neutrons consist of nerve cells, cell body, schema, and precondition depends on the axon, any axis cylinder, nerve fibers. Then parts of neuron, initial substance. These are granules that are stained with the basic dyes like methylene blue, crystal violet, or thionine. Number and size of initial granules vary in cells depending upon its physiological conditions. Few physiological changes like action of some poison or poisonous or to fatigue. The results in discussion of initial granules to fine powders which disappears. These granular component crystals of ribose nucleoprotein RNP, which are responsible for their basic staining property. Then Golgi body, this is the membranous structure like Golgi apparatus in other cells. Mitochondria are rod-like or spherical structure in the cytoplasm of the cell. Then dendrites, these are branches emerging from the main body of the cell and contain mitochondria. The cell granules and neutrophils are as shown in the figure. Then axon, nerve of the fiber extension from the power part of the neuron called axon helix that contains initial granule. A myelinated nerve fiber consists of the following structure: exoplasm and axons. Myelogenesis begins. What is myelogenesis? Myelinogenesis begins with Schwann cells or growing around axon 2 completely enveloped. Axon is surrounded by the cell membrane of Schwann cells, double mesoaxon. Then these cells, each membrane consists of two lipid layers sandwiched between layers of protein. They form myelin sheath of the nerve fiber. Thickness of myelin depends upon the number of layers around axon. Then neurolima. This is outermost cell membrane of Schwann cells and is known as Schwanns of Schwanns. Inside it is present Schwann's cell cytoplasm and its peripheral nucleus. Surrounding is peripheral neural trunk which form sheath of hen, endoneurium. Unmyelinated nerves. These nerves are enclosed by Schwann cells and are not enclosed in myelin sheath. All postganglionic fibers of autonomous nervous system are unlimited as their fibers are somatic nervous system. Then what is the characteristics of the nerve fiber? A nerve fiber consists of a number of neurons, nerve cells, and their function is conduct to nerve impulse. A nerve fiber can be stimulated to any accessible point. Nerve impulse set at any instant travel just as recently as distally. Nerve impulse is generated in nerve cells and travels distally along axons. Its main exemptions are afferent neurons, dorsal nerve roots, and different cranial nerves. In these impulse is set up by appropriate stimulate of sensory terminals. Thank you.